body fluid and circulation. Body fluids பத்தி பார்க்கறதுதான். So we have to see from the smallest living creature in this world to like human being and bigger creatures. Okay. So let us start with simple organism. Simple organisms such as sponges and coelentrates have simple body organization. Their body organization itself simple. In one cell you will have everything: digestive system, respiratory system, circulatory system, everything. Okay. These are in direct contact with atmosphere. Thus, they do not require any circulatory system. They will not have any specific circulatory system. They include circulating water from their surroundings into their body cavities. This facilitates the cells to exchange gases and excretory substances with immediate surroundings. In more complex organisms, special fluids are used to transport these substances within the body. Blood and lymph are two types of fluids that act as a carrier in the body. Blood is a special connective tissue comprising of fluid matrix, plasma and formed elements. Plasma forms the fluid medium in which the blood cells float and carry out different important functions. And they carry out many important functions. Now let us look at plasma. It is a straw colored, light yellowish color, straw color, dirty yellow color, viscous, slightly alkaline aqueous body fluid. It forms about 55% of the blood. Okay. It is composed of many organic and inorganic substances. It contains 90 to 92% water and 6 to 8% solutes in it. The solutes found in plasma are various ions like sodium, magnesium, calcium, bicarbonates, etc. Glucose, traces of other sugars, plasma proteins, amino acids, hormones, cholesterol and other lipids, urea, other waste and other organic acids. Factors for clotting and coagulation is also present. Of the blood are also present actively in the plasma. Plasma without the blood clotting factors is called serum. Plasma ko serum ko, that is the difference. Without blood clotting factor is called a serum. Proteins found in the plasma are important as they are responsible for providing viscosity to the plasma. The major proteins found in plasma are fibrinogen, globulins and albumins. Fibrinogen, globulins and albumins. In the next slide, it performs various functions in the blood. These are: number 1, they help in transport and uniform distribution of heat all over the body. The body temperature is uniform. They distribute heat all over the body. Number 2, it provides body immunity. Next, maintenance of the blood pH. Helps in prevention of blood loss. Fibrinogen helps in blood clotting. Globulin helps in defense mechanism. Albumin maintains osmotic balance. The formed elements are blood corpuscles include erythrocyte, leukocyte and platelets. Erythrocyte, leukocytes and platelets. These constitute about 45% of the blood. 45% of the blood. Erythrocyte, that is red blood corpuscles. These are the most abundant of all cells found in the blood. They are the most abundant of all cells found in the blood. They are red in color due to the presence of pigments called hemoglobin. They are red in color because due to the pigments called hemoglobin, which acts as an oxygen carrier. The formation of RBC takes place in the red bone marrow in adults. Let's look at the shape, size and structure of RBC. They are biconcave disc-shaped cells with the diameter of about 7 to 8 microns. 7 to 8 microns. The shape of RBC is slightly variable as there are no cell organelles found in it. Whole volume is filled with hemoglobin, and the whole volume is filled with hemoglobin. A healthy individual has about 12 to 16 grams of hemoglobin in every 100 ml of blood. It can vary between 12 to 16 grams of hemoglobin in every 100 ml of blood. In men, the average number of RBC is about 5 to 5.5 millions per cubic millimeter. In women, the average number is about 4 to 4.5 millimeter cube of blood. Lifespan of RBC, that is total lifespan of red blood cells, is 120 days, after which RBC becomes non-functional and gets destroyed in spleen. So, spleen acts like a graveyard of RBC. Now let's move on to leukocytes. Leukocytes is the white blood cells. They are known to be the active and motile constituents of blood as well as lymph. They do not possess the red color pigments, and they do not possess the red color pigment, that is hemoglobin, in them. So they are colorless in nature. These are nucleated and are generally short-lived cells. The number of WBCs are relatively lesser in number, about 6,000 to 8,000 millimeter cube of blood. And they move in an amoeboid fashion. These can squeeze through capillary walls and move to the site of action. This phenomenon is called as diapediasis. Diapediasis. Leukocytes or white blood corpuscles are categorized into two main categories. One is the granulocytes. I repeat, it is called as granulocytes. They contain granules in the cytoplasm and have regularly lobed nucleus. Okay, regularly lobed nucleus. The granulocytes are subdivided into three main types. The first one is called as neutrophils. These are the most abundant cells of the total WBC. They stain equally well with acidic as well as basic dyes because they are neutral in nature. These are phagocytic cells that destroy foreign organism entering the body. Destroy the foreign organism entering the body. Next, eosinophils. They are characterized by their bilobed nucleus. Bilobed nucleus. They are stained bright red in color with acidic dye such as eosin. They are about 2 to 3% of total WBC. 2 to 3% of total WBC. They resist infections and are also associated with all allergic reactions. They also help in dissolving blood clots. They help to destroy the toxic substances present in the body. Destroy the toxic substances present in the body. During allergic conditions, the number of eosinophils increase in the body. When you have an allergic condition, the number of eosinophils increase in the body. Now let us look at the basophils. They contain fewer coarse granules than the eosinophils and can be stained with basic dyes such as methylene blue. It can be stained with just a basic dye called methylene blue. They are found least abundantly among WBC. They secrete histamine, serotonin, heparin, etc. and are involved in inflammatory reactions. Inflammatory reactions. Now let's see what is agranulocytes. Agranulocytes, as the name says, it lacks granules in their cytoplasm and have rounded or oval nucleus. Agranulocytes are also further subdivided into two main types. One is lymphocytes. They are smaller in size and have rounded nucleus. Lymphocytes are of two further types. Okay. So agranulocytes is two types. Of that, lymphocytes has got two types. One is B cell and the T cell. Both of these B and T cells are responsible for immune response of the body. So now we have studied the lymphocytes. Let us move on to monocytes. These are largest of all types of monocytes and are known as macrophage. They help to kill foreign particles. These are phagocytic in nature. Now we have finished the WBC. Let's move on to blood platelets. They are called as thrombocytes. These are cell fragments produced from megakaryocytes. That is, the special cell found in the red bone marrow. The special cell found in the red bone marrow. Structure. These are oval-shaped disc-like cells found only in mammalian blood. These are devoid of nuclei. Platelets contain mitochondria, Golgi bodies, and some other structures such as granules, tubules, filaments of actin and myosin. ADP is also present. Let's look at what is the numbers you will have platelets. Blood normally contain 1.5 into 4 zeros. Right? 3.5 to 4 zeros. Platelets per cubic meter. Okay? A reduction in their number can lead to clotting disorders, which will lead to excessive blood loss from the body if someone gets injured. Let's look at the lifespan of platelets. The lifespan of platelets is only 7 to 12 days. Just 1 week, 7 to 12 days. The formation of thrombocytes is called thrombopoiesis. It is called as thrombopoiesis. The next slide. The main function in the body is to release factors, most of which are involved in coagulation or clotting of the blood. Differences between red blood cells and white blood cells. The next slide, the plasma membrane of RBC contains certain glycoproteinase molecules known as antigens. Depending on the nature of antigen, two important common types of blood grouping can be found in the human being. It is the blood group we are talking about. Number one, ABO grouping. It is based on the presence or absence of antigen A or antigen B on the surface of the RBC. People with blood group A have the antigen A on the surface of RBC and have antibodies against antigen B in their plasma. While in the people having blood group B, the case is just vice versa to blood group. It is exactly opposite. Okay. A, B and O blood groups were discovered by Landstein. Blood group AB was discovered by D. Castillo and Staining. In people with blood group O, both antigen A and B are not present on their RBC, but they have antibodies against in the plasma. This distribution of antigen and antibodies in the four blood groups A, B and A, B, AB and O. Next we have the RH grouping. Apart from the antigen discussed, another antigen known as RH antigen is also found on the surface of the RBC in majority of human being. Majority of the human being. Individuals having RH antigens are RH positive, and those without RH antigens are called as RH negative. Very interesting. So you will know a lot about this as we move on. Okay, let's move on to the next slide. A special case of mismatching of RH group or RH incompatibility has been observed between the RH blood of pregnant mother with RH blood of the unborn baby inside the tummy. It is called as fetus. The fetus of RH woman and RH man. Okay. So if the woman is negative, man is positive, what will happen? In such a case, mother becomes sensitive carrying an RH positive baby in the womb. The reason is that some of the RBCs from developing baby and developing fetus enters some of the RBCs enters the bloodstream of the mother during development. Okay, what will happen? So baby's RH positive enters mother's RH negative. Baby has antigen, mother does not have antigen. This causes the development of anti-RH antibodies in mother's blood. The RH antigen of the unborn baby, that is fetus, do not get exposed to RH negative blood of the mother in cases of first pregnancy. However, during delivery of the first child, there occurs a possibility that maternal blood may get exposed to small amount of RH positive blood from the fetus, hence preparing antibodies against RH antigen in her blood. Thus, in case of her subsequent pregnancies, RH positive fetuses get exposed to anti-RH antibodies. These antibodies will reach the blood of the fetus, that is unborn baby, that is RH positive, and destroy the baby, destroy the fetus, RBCs, destroy the baby's RBCs. This could be fatal to the developing fetus and could cause severe anemia and jaundice to the unborn baby, the developing baby, the fetus. This is known as erythroblastosis fetalis or hemolytic disease of the newborn. Hemolytic disease of the newborn, or you can call it as unborn. In this condition, can be avoided by administering anti-RH antibody to the mother immediately. This is the rhesus incompatibility which is based on. So we are talking about rhesus incompatibility. So when you give antibodies, the baby will be protected, mother will be protected immediately after the delivery of the first child. RH factor was discovered by Landsteiner and Vanier after immunizing the rabbits with the blood of a monkey. There are 30 plus antigens on the surface of red blood cells that give rise to different blood groups. They are MN blood group. There are various different types depending upon the antigens. Okay. Now let's move on. In the next slide, when an injury is caused, the wound normally does not continue to bleed for a long time and the blood stops flowing after some time. It is natural property exhibited by the blood to check the excessive loss of blood from an injury or trauma. Clotting of the blood is a complex process that involves various enzymatically controlled steps for its completion. Enzymatically controlled steps for its it is completion. Okay. Here an injury or a trauma causes stimulation of the platelet cells or release of certain platelet factors. This in turn activates the mechanism of coagulation or clotting of the blood at the site of injury. There are 3 steps we have to remember. The first step is the thromboplastin. Remember the name thromboplastin helps in formation of an enzyme prothrombinase that converts the inactive plasma protein, that is prothrombin, into its active form called as thrombin. Prothrombin into its active form called as thrombin. Thrombin thus acts as a proteolytic enzyme to convert fibrinogen molecules to form insoluble fibrin monomers. This reaction required thrombokinase, an enzyme complex which is formed by a series of linked enzymatic reaction which involves a number of various factors present in the plasma in their inactive state. Both the changes mentioned above requires calcium ions for their reactions. Remember, calcium ions. These fibrin monomers polymerizes to long sticky fibers. The fibrin threads form a fine network of threads called fibrins, in which dead and damaged formed elements of blood are trapped. This finally leads to the formation of a clot or coagulum, which is a dark reddish brown scum formed over the surface of the injury. Okay. Here in this slide, the blood performs few important functions, and let us see one by one. Okay. The blood performs few important functions. Number 1, it helps in transportation of respiratory gases. Number 2, helps in healing of wounds. Number 3, maintains body pH. They maintain the body pH. 4 is water and ionic balances. Next, fights against infection by forming body immunity. Also helps in transportation of hormones from endocrine glands to target organs. Next, coagulation of the blood. Then helps in transportation of body waste from different body parts to the kidney. Then maintains normal body temperature. So these are the different functions. Okay. It is another fluid connective tissue that floats inside specialized vessel known as lymph vessels. We are talking about lymph here. Composition. It is colorless fluid containing high concentration of WBC. The overall composition of lymph is similar to blood with the exception of absence of RBCs, platelets and some plasma proteins, and in having less calcium and phosphorus. Having less calcium and phosphorus than their levels in the blood. It also contains all the ions present in the blood plasma. In the next slide. It is an elaborate network of vessels which collects the interstitial fluids. The next slide. As the blood passes through the capillaries of arterial system into the tissues, some water along with many water-soluble substances come out in the spaces between the cells of the tissues. But a very small amount of protein come out from the capillaries with the plasma. Protein, it will be very small amount. The fluid thus released out is called interstitial fluid. After entering the lymph vessels, the extracellular fluid becomes lymph. The extracellular fluid becomes lymph. That's how the lymph is formed. Interesting, isn't it? Great. As we know, blood and lymph are important fluids in the body that act as a carrier in transporting nutrients, hormones and various other materials even to the farthest tissue in the body. Now, the question arises, what makes this blood to circulate inside the body? What makes this blood to circulate inside the body? So, the answer to this question lies in the fact that to circulate this blood throughout the body, a muscular pump is required, which is known as heart. The heart we talk about is different. Here it is a muscular pump. Make sure blood is circulated all over the body. Depending upon the circulatory pattern in higher organism, the circulatory system is of two types. One is the open type, open circulatory system. Another one is the closed type. Open type, it is the type of circulatory system in which blood is pumped by the heart, is passed through a vessel into spaces or body cavities. The body cavities is known as sinuses, from where the blood is collected by large vein which opens into the heart. In this type of system, the blood does not contain the respiratory pigment and is present in dissolved in plasma. It is present in the dissolved in plasma. This type of system is found in arthropods and molluscs. There is no capillary system found in this type of circulatory system. Next, look at closed circulatory system. Closed circulatory system. Here, the circulatory system, the blood is pumped by the heart, is always circulated in a closed vessels. From evolutionary point of view, if you look at the evolutionary point of view, this pattern is considered to be most advantageous as it supplies blood to the deepest tissue of the body. Supplies to the deepest tissue of the body. It is found in annelids and chordates and even human being. So the next slide, depending upon the different types of circulation in them, heart is three types. Okay. So there are animals which has got two-chambered heart. Just two chambers. This is found mostly in fishes, mainly fish. It is comprised of atrium and ventricle. Just atrium and ventricle. During circulation of blood, the heart pumps out deoxygenated blood. This blood gets oxygenated by the gills and is then supplied to body parts from where the deoxygenated blood returns to the heart. Deoxygenated blood returns to the heart. Hence, the type of circulation is called single circulation. It is called just single circulation. Next, we have the three-chambered heart. We have seen two, now it is three-chambered heart. Amphibians and reptiles have a three-chambered heart. This type of heart comprises of two atria and a single ventricle. Two atria and a single ventricle. During the circulation of blood, the left atrium receives the oxygenated blood from the gills or lungs or skin, and the right atrium receives the deoxygenated blood from the other parts of the body. The blood gets mixed up in single ventricle which further pumps out the mixed blood. This type of circulation is called incomplete double circulation. It is called as incomplete double circulation. Next, we'll see four-chambered heart. We've seen 2, 3 and now it's four-chambered heart. Crocodiles, birds and mammals, including human beings, possess this type of heart. It comprises of two atria and two ventricles. During the course of circulation, the oxygenated and deoxygenated blood is received by the left and right atrium. This is left and that's right atrium respectively. The blood from each atria is passed on to the ventricle of same side. So left comes to the left side, right comes to the right side. Okay. This is called double circulation. There is no mixing of oxygenated and deoxygenated blood and both flow simultaneously in heart chambers but separately. So deoxygenated blood comes in and they flow separately. Oxygenated blood comes in and they flow separately. So it is called a double circulation. In the next slide, circulatory system in human is also known as body vascular system. It consists of muscular chambered heart, a network of closed branching blood vessels, and the fluid that is circulated, that is blood. The fluid that is circulated is called blood. Moving on to the next slide, heart is a mesodermally derived organ situated in the thoracic cavity in between the two lungs. It appears to be slightly tilted towards the left side. It is a hollow fibromuscular organ, slightly conical in shape, of about 12 cm length and 9 cm breadth. The upper broad part is called the base and the lower narrow part known as apex. Lower narrow part is called as apex. It has a size of a clenched fist. This is clenched fist. Covering the heart is protected by a two layers, double-walled membrane sac or a bag called pericardium. The fluid present between two membranes is called pericardial fluid. This fluid helps in keeping the surface of the heart moist and also protects it from shock and mechanical injuries. Structure of human heart can be studied under two headings. Let us see what is that. First is external and second is the internal structures. Okay. In the next slide, we will be studying about the external structures. Externally, the human heart is composed of four chambers. Two relatively smaller upper chamber called auricles and two larger lower chambers called ventricles. The right atrium is slightly larger than the left atrium. Both these atria are meant to receive blood from different body parts. Next, we'll study about the internal thing. Internally, the chamber of the heart, the two auricles and ventricles are separated by different septa and valves. There is different septa and valves. In this slide, what we are going to see is upper two thin-walled and smaller chambers. They serve to receive the blood, therefore are called receiving chambers. Both the right and the left atria are separated by a thin muscular wall known as internal septum, right atrium. The right chamber deals with only impure blood It receives impure blood from various parts of the body through two major veins, just two major veins. The first one is the superior vena cava and the second one is the inferior vena cava. Inferior vena cava. Now look at the left atrium. This chamber is meant to deal with only pure blood, It receives pure blood from lungs through two pulmonary veins. Remember that it is pulmonary veins. Here there are lower two chambers of the heart that pumps the blood away from the heart. This functions as pumping chambers. Both the right and the left ventricles are separated by the interventricular septum. It is called as interventricular septum. The atrium and the ventricle of the same side are also separated by another septum, a thick fibrous tissue called atrioventricular septum. It is called as atrioventricular septum. Right ventricle, it receives impure blood from right atrium and pumps blood into pulmonary artery. It goes to the impure blood, pumps into pulmonary artery, which further takes this blood to the lungs for purification. Next, left ventricle, it receives pure blood from left atrium and it pumps pure blood to aorta, which in turn takes the blood to whole body and organs. Here in this slide, apart from septum, heart is also separated by various valves. These valves act as a door-like structure in the heart that serves to maintain the unidirectional flow of blood. Different valves present in the heart are: number one, tricuspid valve. It is formed by three muscular flaps or cusp to guard the opening between the right atrium and right ventricle. Next, bicuspid valve, that is mitral valve. Bicuspid valve. Tricuspid, now this is bicuspid. It is the type of valve that guards the opening between the left atrium and left ventricle. Next is semi-lunar valve. The opening of right and the left ventricles into the pulmonary artery and the aorta are provided with the semi-lunar valves. Now look at the function of the cardiac valves. There are three valves, we have studied about it. What is the function of them? The valves in the heart allow the flow of the blood only in one direction, that is from atria to ventricle and from ventricle to pulmonary artery or aorta. Okay, and prevents any backward flow of blood into four chambers. There is no backward flow normally. Next, the heart is made up of cardiac muscles. The ventricular walls are thicker than that of the atrial valves. The rhythm of heart is maintained by a highly specialized cardiac musculature called the nodal tissue. It is called as nodal tissue, distributed evenly in the heart muscles. Next, Cyanoatrial node or SA node. The SA node is a small flattened patch of specialized tissue present in the right upper corner or right atrium. The impulse generated by this node spreads in all directions of the heart. It spreads in all directions of the heart. Next, the Atrioventricular Node or AV node, the signals which arise from SA node becomes weak when they reach ventricles, because ventricles are far away from SA nodes. Ventricles are far away from SA nodes. Thus, to strengthen these signals, another mass of tissue is seen in the lower left corner of right atrium, close to the atrioventricular septum, known as AV node. It is known as AV node. This is also known as pace setters. It is also known as pace setters. Third, bundle of His. It is called as bundle of His. A bundle of nodal fibers, that is atrioventricular bundle, AV bundle, continues from the AV node, which passes through the atrioventricular septum to emerge on the top of the interventricular septum. Top of the interventricular septum, immediately dividing into a right and left bundle. Okay, so it's a right bundle and a left bundle. This bundle gives rise to a network of minute fibers throughout the ventricular musculature of respective side, known as Purkinje fibers. It is called as Purkinje fibers. These fibers, along with the right and left bundles, are called bundle of His. The Purkinje fibers conduct impulses to all portions of ventricular wall of the heart. To all portions of ventricular walls of the heart. The next slide. The nodal musculature possesses the ability of generating action potentials without any external stimuli. That is, it is auto-excitable. However, the nodal system generates different number of action potential at different parts in minute. In a minute, its action potential is in the different parts. The SA node can generate the maximum number of action potential, that is 70 to 75 per minute. 70 to 75 per minute. It is also responsible for the initiation and maintenance of the rhythmic contractile activity of the heart. Therefore, the SA node is also called pacemaker. It is also called as pacemaker. Our heart normally beats 70 to 75 times in a minute. That is, average 72 beats per minute. Okay. Moving on to the next slide. As man has closed circulatory system, the blood strictly flows in fixed route through arteries and veins, maintains a continuous flow throughout the body inside the closed tubes or blood vessels. These tubes or vessels are of mainly two types. Okay, we all know these things, isn't it? These blood vessels carry blood from heart to different parts of the body. They are called as arteries. All the arteries carry pure oxygenated blood with the exception of pulmonary artery. Apart from our pulmonary artery, all the arteries carry pure blood. Pulmonary artery carries impure blood or deoxygenated blood. Okay. As the walls of arteries are thick and are non-collapsible, the pressure inside them is very high. Okay. Let us look at the salient features of arteries. Okay. Valves are absent in arteries. There is no valves. Arteries are divided into fine branches known as arterioles, which are further divided to form finer branches called capillaries. Now let's see veins. These are another type of blood vessels that bring blood from different parts of the body to the heart. They carry deoxygenated blood to the heart, dirty blood. Okay? They carry blood towards the heart. All veins are meant to carry impure blood except pulmonary vein. We studied with pulmonary artery, this is pulmonary vein. The pulmonary vein that carries pure blood from lungs to the heart. Veins are provided with valves to prevent backward flow of the blood. So veins have valves. We just remember that. Okay. Next, the heart pumps the blood to all parts of the body. The changes that takes place in the heart during one heartbeat together constitute cardiac cycle. Okay. That simple is cardiac cycle. The heartbeat at an average rate of 72 per minute. Thus, total duration of cardiac cycle is just 0.8 seconds, less than 1 second. During a heartbeat, the contraction and relaxation of atria and ventricles takes place. The phases of contraction is known as systole, while the relaxation phase is called as diastole. Thus, a single heartbeat consists of systole and diastole of both atria and ventricle. To begin with, the cardiac cycle, all four chambers of the heart are relaxed. They are in joint diastolic, during which the blood flows from the superior and inferior vena cava into atria and from there to respective ventricles through atrioventricular valves, through auriculoventricular valves. The complete cardiac cycle is comprised of certain events that takes place in a sequential manner. Here in this slide, a wave of contraction occurs from anterior to posterior side stimulated by the SA node. The blood flows from the pulmonary veins and vena cava into left and right ventricles respectively, as the tricuspid and bicuspid valve are open. During this time, the blood does not return to great veins. The semilunar valves are closed. The atrial systole increases the flow of blood into the ventricles by about 30%. At the end of the atrial systole, there starts relaxation of atria and contraction of the ventricles simultaneously. The atrial systole occurs for 0.1 seconds, while the atrial diastole, on the other hand, is of about 0.7 seconds. In the next slide, this step involves simultaneous relaxation of atria and contraction of ventricles. As the contraction of ventricles begins, the process of blood rises in them almost immediately. This rapidly closes the atrioventricular valves in order to prevent the backflow of blood from ventricles to atria. The conduction of action potential to the ventricular side occurs by the AV node and AV bundles, from where the bundle of His transmit it through the entire ventricular musculature. The contraction of ventricles thereby tricuspid and bicuspid valve due to attempted backflow of blood into the atria. Finally, due to this, the increase in the pressure occurs in the great arteries. So, semilunar valves guarding the pulmonary arteries and aorta are forced open and blood enters through great arteries into ventricles. Okay? When ventricles relax, the ventricular pressure fails, which causes the closure of semilunar valves, preventing the backflow of blood into the ventricle. The further decline in the ventricular pressure opens the tricuspid and bicuspid valves by the pressure in atrial exerts by the blood, which was being emptied into them by the veins. This allows the blood to move freely to the ventricles. Once again, the ventricles and atria are now again in a relaxed state as earlier. Soon, the SA node generates a new action potential and the events described above sequentially repeats to continue this process. Sequentially continues this process. Okay. Moving on to the next slide, whatever we have seen is, we have studied that heartbeat for about 72 times per minute. This concludes that in a single minute, many cardiac cycles are performed, thus reducing that duration of each cardiac cycle is 0.8 seconds. During each cardiac cycle, each ventricles pump out about 70 milliliters of blood. This is known as stroke volume. This is known as stroke volume. The volume of blood pumped out by each ventricle in 1 minute is called as cardiac output in 1 minute. We know that in 1 minute heart beats for 72 times. Thus, cardiac output will be 540 milliliters or approximately 5 liters in a normal individual. Thus, cardiac output equals stroke volume and number of beats per minute. It is to be noted that the body has the ability to alter stroke volume as well as the heart rate and thereby the cardiac output. Athletes have much higher cardiac output than the normal man. Okay. In the next slide, during each cardiac cycle, two prominent sounds are produced which can be easily heard by a stethoscope. This allows to hear sounds and pulses of an individual. The basic reason for the production of these sounds is the closure of various valves. The sounds cardiac produced during each heartbeat, first is lub, 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 lub. It is the first sound being produced when interauriculoventricular valves are closed. This marks the end of atrial systole and beginning of ventricular systole. Next is dub, 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 dub. It is the second sound being produced when semilunar valves are closed. This marks the end of ventricular systole. In the next slide, it is a graphical representation of the electrical activity of heart during single cardiac cycle. The electrocardiogram is obtained by a machine known as ECG machine or electrocardiograph machine. The study or the process of recording of the electrocardiogram is called as electrocardiography. The impulse generated by a SA node causes contraction and relaxation of the heart chambers. To obtain a ECG, a patient is connected to the machine with three electrical leads monitoring the activity of the heart continuously and heart functioning evaluated by attaching multiple leads to the chest region. Multiple leads to the chest region. In the next slide, an ECG consists of 5 peaks identified with the letter P to T that corresponds to a specific electrical conductivity of the heart. These corresponds to a specific electrical activity of the heart. First is the P wave. It is the first and the foremost wave of low amplitude. It represents the electrical excitation or depolarization of atria, which leads to contraction of both the atria. So that is P wave. Next is QRS wave or QRX complex. The Q, R and S wave together forms the QRS complex. This represents the depolarization of the ventricles, which initiates the ventricular contractions. It marks the spread of impulses from AV node to the ventricles, through bundle of His and Purkinje fibers. The contraction starts shortly after Q and marks the beginning of the systole. Lastly, the T wave. It is a broad and smoothly round deflection which represents the return of ventricles from excited to normal state. The end of T wave marks the end of systole. It has been observed that by counting the number of QRX complexes that occurs in a given time period, one can easily determine the rate of heartbeat of an individual. However, the deviation in the ECG of any person from normal shape ECG indicates a possible abnormalities of a disease. Right? It indicates that there is possibility of the abnormality. Okay, let's see the next slide. Right? ECG also proved to be of great clinical significance due to what are the reasons? Let's see it. It gives accurate information about thermal functioning of atria and ventricle. Number 2 indicates the functioning of valves. Next, also helps in indicating any damage to local tissues of the heart in detection of growth of cardiac and heart chambers. Next, what you're going to see is the double circulation. Okay, the mammalian heart is 4-chambered, undergoing the process of complete separate double circulation. This means that the blood passes twice through the heart to supply the blood for once to the body. This transmission is necessary as it helps in oxygenation of the blood. Number 1, pulmonary circulation. In this system, blood completes its circulation from right ventricle to left atria through the lungs. Here, the deoxygenated blood pumped by the right ventricle enters the pulmonary artery, while the left ventricle pumps blood into aorta. The deoxygenated blood pumped by the right ventricles enters the pulmonary artery, while the left ventricle pumps blood into the aorta. The deoxygenated blood, I repeat, the deoxygenated blood is passed on to lungs, from where the oxygenated blood is carried out by the pulmonary vein into left atrium of heart. Next, systemic circulation. In this system, the pure blood is supplied to all parts of the body. During transport, the oxygenated blood entering the aorta is carried by a network of arteries. Network of arteries, arterioles and capillaries to the tissue from where deoxygenated blood is collected by a system of veins and vena cava, thus emptying it into right atrium. This system provides essential nutrients, oxygen and other essential substances to all the tissues of the body and eventually take away carbon dioxide, other harmful substances away from the tissue, and then they are eliminated from the body. Apart from the pulmonary or systemic circulation, heart also possesses other circulatory pathway. Hepatic portal system. It is the type of portal system which is universally present in all vertebrates. It is a unique vascular system that exists between the digestive tract and liver. A vein called hepatic portal vein carries blood from the intestine to the liver. The liver finally opens into inferior vena cava through its own veins before delivering it to the systemic circulation. Simple, isn't it? Yeah. In the next slide, apart from hepatic portal system, another minor portal system called hypophyseal portal system. It is also present, which consists of two hypophyseal veins, that is in the brain, midbrain. Hypophyseal portal vein which carries blood from the hypothalamus to anterior to the pituitary gland, anterior lobe of the pituitary gland. Okay. Next, although the heart is always filled with blood, it does not take its oxygen and nutrients from the blood present in the chambers. Thus, in order to supply oxygen and nutrients to the muscles of the heart, a cardiac system is present, so highly advanced. Coronary system is present. The right and left coronary arteries arise from the ascending aorta, which supplies oxygenated blood to the heart muscles. The coronary veins bring deoxygenated blood to coronary sinus, thus carries it to the right atrium. Okay. In the next slide, the normal activities of the heart are intrinsically regulated very minutely. First, auto-regulated by the specialized muscle. Thus, the heart is known as myogenic heart. The neurogenic heartbeat is initiated by a nerve impulse. For example, annelids and most arthropods. The rate of formation and conduction is regulated by, first, neural regulation. In medulla oblongata, a special neural center is present, which can moderate the cardiac function through the autonomic nervous system. The neural signals through the sympathetic nerves increases the rate of heartbeat by stimulating SA node. It also increases the strength of ventricular contractions and thereby the cardiac output. Increases the ventricular contractions, thereby the cardiac output. Whereas the neural signals through parasympathetic nerves can decrease the rate of heartbeat by inhibiting the SA node spread of conduction of the action potential and thereby decreasing the cardiac output. Next, look at the hormonal regulations. Okay. Adrenaline and noradrenaline hormones secreted by the medulla of adrenal glands has significant role in regulating heartbeat and thus increasing the cardiac output. The noradrenaline accelerates the heartbeat while adrenaline does this function at a time of emergency, only at the time of emergency. Let us look at the diseases of the heart. Many diseases may affect the blood vessels and the normal functioning of the heart. Some of the common disorders are, yes, high blood pressure, which is called as hypertension. The pressure exerted by the flow of blood on the elastic walls of the arteries is known as blood pressure. Hypertension is the term used for blood pressure higher than the normal. The normal blood pressure in human is measured to be 120 over 80 millimeters of mercury, in which 120 millimeters of mercury is the systolic pressure, while the 80 millimeters of mercury is the diastolic pressure. It is called the diastolic pressure. Persistent increase in blood pressure above 140 millimeters of mercury and 90 millimeters of mercury is termed as hypertension. Above 140 and 90. It is termed as hypertension. Conditions of hypertension. It may lead to many heart disease and also affect vital organs of the body like the brain, kidney and various organs. Next, look at the coronary artery disease. We have seen hypertension, now it's a coronary artery disease. It is the hardening of the arteries and arterioles due to the thickening of the fibrous tissue and the consequently loss of elasticity. It is often referred to as atherosclerosis. This mainly affects the vessels, which are mainly responsible for supplying blood to the heart muscles. It seems to occur due to deposition of calcium. Deposition of calcium, deposition of fat, cholesterol and other fibrous tissue, making the lumen of the artery narrower. Next, let us see angina. It is also called as angina pectoris because it's in the pectoral region. This tends to occur when enough oxygen does not reach the heart muscle. It occurs both in men and women of any age but seems to be more common among the middle-aged and elderly individuals. A system of acute cardiac chest pain occurs in individuals suffering from angina, which mainly occurs due to the condition that affects the blood flow. Next, look at the fourth one, the heart failure. Heart failure. It is the condition of the heart when it fails to pump blood effectively to meet the needs of the body. The heart failure is sometimes called as congestive cardiac failure or congestive heart failure, as its main symptom is congestion of lungs. The lungs gets congestion. Next, look at this. What is bradycardia and tachycardia? Tachycardia is term applied to a rapid heart or pulse rate. Bradycardia is the term indicating a slow heart or pulse rate. That is under 50. Okay. These are the main symptoms. Of course, there are many other things. When you become a doctor, you will learn those things. In MBBS, you learn certain diseases. In MD physiology or MS surgery, you learn much more. After that, if you do open heart surgery or intervention cardiology, you will be doing a different one. You will be reading much more. All the best. That's over this chapter. Excellent, kids.